Many of life's little chores can be quietly satisfying and rather fun. Washing dishes by hand, however, is not so fun, which is why some clever person made the dishwasher. This handy invention soaks, washes, and rinses your dishes to a spot free, sanitary sparkle. You unload the dishes, and the machine is ready to start the cycle all over again. A cell goes through a cycle too. This cell is growth, DNA synthesis, and division, and they are essential for an organism to grow and heal. If it goes out of control, abnormal cell growth may occur, resulting in cancer cells like the ones shown on the screen here. Chapter 5.1, The Cell Cycle. Key concept. Cells have distinct phases of growth, reproduction, and normal functions. Main ideas. The cell cycle has four main stages. Cells divide at different rates, and cell size is limited. First main idea. The cell cycle has four main stages. Just as all species have life cycles, from tiny chihuahuas to massive beluga whales, cells also have a life cycle. The cell cycle is the regular pattern of growth, DNA replication, and cell division that occurs in eukaryotic cells. Figure 1.1 on the screen here shows its four main stages: gap 1, synthesis, gap 2, and mitosis. Gap 1, synthesis, and gap 2 together make up what is called interphase. The stages of the cell cycle get their names from early studies of cell division. Scientists' observations were limited by the microscopes of the time. When a cell was not actively dividing, they could not see activity in it. Thus, they originally divided the cell cycle into two parts: interphase, when the cell appeared to be at rest, and mitosis, when the cell was dividing. Improved techniques and tools later allowed scientists to detect the copying of DNA, which is called DNA synthesis, and they changed their description of the cell cycle to include the synthesis stage. Since they could still not see anything happening during the other parts of interphase, scientists named the periods between mitosis and synthesis gap 1 and gap 2. Eventually, scientists learned that, during interphase, cells carry out their normal functions and undergo critical growth in preparation for cell division. The first stage of the cell cycle is gap 1, also known as G1. During G1, a cell carries out its normal functions. If it is a skeletal muscle cell, it contracts to move joints. If it is an adrenal cell, it secretes hormones such as adrenaline. If it is an intestinal cell, it absorbs nutrients. During G1, cells also increase in size, and organelles increase their number. The cell responds and spends most of its time in the G1 stage, although the length of this stage varies by cell type. During G1, the cell must pass a critical checkpoint before it can proceed to the synthesis stage. Just as it would be dangerous for you to run a marathon if you had not slept or eaten for several days, it would also be dangerous for your cells to continue dividing if certain conditions were not met. For instance, most animal cells need enough nutrition, adequate size, and relatively undamaged DNA to divide successfully. They also need specific signals from other cells telling them whether more cell division is needed. The second stage of this cell cycle is the synthesis, or S stage. Synthesis means the combining of parts to make a whole. During the S stage, the cells make a copy of its nuclear DNA. In eukaryotes, DNA is located in the nucleus. During interphase, it is loosely organized and appears grainy in photographs. By the end of the S stage, however, the cell nucleus contains two complete sets of DNA. Gap 2, also known as G2, is the third stage of the cell cycle. During G2, cells continue to carry out their normal functions and additional growth occurs. Like G1, this stage includes a critical checkpoint. Everything must be in order: adequate cell size, undamaged DNA, before the cell goes through mitosis and division. Mitosis, also represented by a capital M, which is the fourth stage of the cell cycle, includes two processes: mitosis and cytokinesis. Mitosis is the division of the cell nucleus and its contents. During mitosis, the nuclear membrane dissolves, the duplicated DNA condenses around proteins and separates, and the two new nuclei form. Lastly, cytokinesis is the process that divides the cell's cytoplasm. The result is two daughter cells that are genetically identical to the original cell. The stages of the cell cycle and the proteins that control it are similar in all eukaryotes. For example, scientists have demonstrated that some of the molecules that regulate checkpoints in the yeast cell cycle can work in human cells too. Such similarities suggest that eukaryotes share a common ancestry. Second main idea: cells divide at different rates. Rates of cell division vary widely, as is shown on the screen here in Figure 1.2. The prokaryotic cycle is similar, but not identical to that of eukaryotic cells. Recall that prokaryotes do not have the membrane-bound organelles and cytoskeleton found in eukaryotes. Thus, prokaryotic cells typically divide much faster than do eukaryotic cells. The rate at which your cells divide is linked to your body's need for those cells. In human cells, the S, G2, and M stages together usually take about 12 hours. The length of the G1 stage differs most from cell type to cell type. The rate of cell division is greater in embryos in children than it is in adults. Children have a shorter cell cycle, and many of their organs are still developing. But the rate of cell division also varies within different tissues of the adult body. The internal lining of your digestive tract receives a lot of wear and tear. As a result, cells that line your stomach and intestines are replaced every few days. In contrast, cells that make up the rest of your intestine, mainly the smooth muscle, and many of your internal organs such as lungs, kidneys, and liver, divide only occasionally in response to injury or cell death. Cells that divide only rarely are thought to enter a stage that some scientists call G0. In G0, cells are unlikely to divide, although they continue to carry out their normal functions. Some cells, such as neurons, appear to stay permanently in the G0 stage. However, some data suggest that neurons actually can divide, and this question continues to be actively researched. Other cells, such as lymphocytes, which is a type of white blood cell, may remain in G0 for years until they recognize an invader. Once the invader binds to a lymphocyte receptor, the lymphocyte goes through rapid cell divisions to help fight infection. Third main idea: cell size is limited. Cells have upper and lower size limits. If cells were too small, they could not contain all the necessary organelles and molecules. For instance, a cell with too few mitochondria would not have enough energy to live. However, cells cannot grow beyond a certain size, even if surrounded by plenty of nutrients. The upper limit on cell size is due to the ratio of cell surface area to volume. Recall that oxygen, nutrients, and wastes move across the cell membrane or the surface of the cell. These materials must be transported in adequate amounts and with adequate speed to keep the inside of the cell functioning. But as a cell increases in size, its volume increases faster than its surface area, as is represented on the screen here in Figure 1.3. Therefore, a further increase in size could result in a surface area too small for the adequate exchange of materials. Some cells, however, must be large. A neuron running down a giraffe's neck to its legs may be several meters long, for instance, but it is not shaped like a cube or a sphere. Instead, it is extremely long and thin. This structure gives the neuron a large surface area with a relatively small increase in volume. To maintain a suitable cell size, growth and division must be coordinated. If a cell more than doubled its size before dividing, the daughter cells would be larger than the original cell. If this happened with each generation, cells would quickly become too large to live. Similarly, if a cell did not double its size before dividing, the daughter cells would be smaller than the original cell. If this happened with each generation, cells would become too small to live. This has been Chapter 5.1, The Cell Cycle. Key concept. Cells have distinct phases of growth, reproduction, and normal functions. Main ideas. The cell cycle has 4 main stages. Cells divide at different rates, and cell size is limited.